Hi everyone, my name is Aisha Jawo, and today I'm going to be talking about neocortical neuronal morphology in newborn giraffes and African elephants. So, I chose this topic because the function of the brain, the different parts of the brain, and their functions have always been interesting to me. So, seeing a study that did this in different mammals stood out to me, as well as the fact that this is a rare opportunity for them to be able to do this in the newborn giraffe cells, and the fact that they just had these tissues that were preserved well available to them. And so before we get started, I'm going to run through a quick outline of what we're going to talk about, some background information and some terms that we're going to be using in this, as well as what the experimenters hypothesized and why they did that, and the methods used, as well as the data or the figures from this and the results that were concluded, as well as the real world correlation. So the first term is a neuron, and this is the working unit of the brain. This is the special cell that transmits information to other nerve cells, muscles, or gland cells, and they do this through using electrical impulses and chemical signals in order to transmit the information between the different parts of the brain and the rest of the nervous system. So over here you see the axon and the cell body, as well as the dendrites, which we'll be talking about a lot throughout this experiment. The next term is morphology, and in biology, this is the study of the shape and the structure of animals, plants, and microorganisms, and their relationship to constituent parts. So, in the study, morphology relates to the shape of the neuron, which the experimenters were interested in. The third term is the neocortex, which is the cerebral cortex that's involved in higher brain order and brain functions such as sensory perception, cognition, and generation of motor commands. And in humans, this is where language comes in. So this study is conducted about neuron morphology in cerebral or the neocortex. So the neocortex is 6 layers, and it's a distinguishing feature in mammals because it's been found in the brain of mammals, but it's not been found in any other animals in the shape and the structure being the same. And it contains billions of neurons. Humans have about 86 billion. And so recent studies have been done about morphology of the neocortical neurons in large brain mammals, and they've revealed that evolution has a large part to play about the conserved features across these species. So, and it relates to characteristics that are unique to um, phylogenetic groups. So, in African elephant neocortexes, the morphology is distinct from all other mammals. That's what they've concluded from the studies, as well as in dendrite morphology as well. So the morphology observed in these species have been limited to adult neocortexes so far. So all the investigations that were done on neomor- neomorphological neurons have all been done on adult mammals. And this is the first study that I've seen done on newborn ones, newborn mammals. And this is because, like I was saying earlier, of the availability of the preserved newborn brain tissue in both elephants and giraffes. And this is where this rare opportunity came from. And it helps to verify the neuron morphological findings that were done in adult elephants and giraffes, but and also to document new findings and come up with new theories and hypotheses about the newborn ones. So next is the hypothesis. In this, the experimenters came up with a few different things. In the dendritic system differences, they expected the dendritic system with the same neuronal types to be more extensive in the elephants than in the newborn giraffes. And dendrites are the branches projected from the neurons. And this is because this same hypothesis has been proven right in adult elephants. That from this study that was conducted, it was about the implications for brain evolution in mammals, and this is where they got this information from, and they hypothesized that it would be the same in newborn elephants rather than in newborn giraffes. And also there were differences between the adult giraffe and elephant brain, but they still expect the results to correlate to that of the study. Second part of it. So since the newborn samples allowed them to investigate the differences, they can compare the adult elephant and the adult giraffe to the newborn elephant and giraffe tissues obtains, and they expected the giraffe neocortex to exhibit a more columnar organization. So this means that they expect the dendrites to have vertical ascending shape more than the newborn elephants, which they expected to have wide apical dendrites. And they also expected a greater variety of neuronal morphology because unlike other animals that have been used or other mammals that have been used for this experiment. And again, the neocortex is the six-layer cortex that's responsible for higher brain functions. The third part was the variation expected. They didn't really expect a lot of variation in some of the neurons present in both of these, such as the inner neurons. And this is because these neurons have been shown to be relatively the same morphologically in all of these mammals. Next is the experimental method. So after these cortical samples were obtained from the newborn giraffe and newborn African elephant, from the experimenters got these from a zoo. They were able to cut out areas of interest and stain it with a Golgi technique. And so the Golgi technique was used in order to prevent experimental bias. And in the giraffe and the elephant, 3 to 5 millimeters uh, thick brain tissue was removed, primarily from the motor cortex and the visual cortices. So in the giraffe, you can see these two rectangles right here. Those are the places where it was removed. This is where the sample removed is. And in the elephant, it was these three. So in this dorsal view picture, you can see that they removed more from the giraffe than the elephant. However, they used the right side of each in order to be able to do that. So this picture shows it done on an adult giraffe and elephant because the newborn ones, their their brains were a bit too fragmented in order to be able to structure it out or provide accurate drawings of what was happening. So instead, they just give the weights over here of each. So in the methods, after they did the Golgi staining and they removed the tissue, they calculated the number of dendrites, the number of neurons, and the dendrite volume, as well as the dendrite length, which is the sum length of all the dendritic segments. And they also took the dendritic spine density, which is the average number of spines of all the dendritic lengths. So, statistical analysis of these two species show a difference between the small number of neurons that were traced in each cortical for each of the species. And as a result, the Golgi snapshot that was provided was only able to include about 84 quantified neurons from the giraffe and 69 from the elephants, even though more more brain tissue was removed from the elephant than in the giraffe. So overall, the newborn elephant neurons had more extensive dendrites, particularly in terms of volume, and they were considered larger in terms of spine measurements as well. So from this, you can see the layer of the cortex, where these two are the little rectangles that were taken out from the giraffe brain, and these three are from the elephant brain. So in the giraffe brain, you can see the motor and the visual cortices. And in the elephant brain is the motor, the visual, as well as the occipital. And so after they looked at the layer, they realized that the frontal cortex, as well as all the other cortexes of the elephant brain, was thickest in all the regions. And even though the giraffe one is already considered thick, the elephant was seen to be this by a large amount. So this is showing, mm, this is showing the different neurons. Um, so the inner neurons, which is B and J over here, B and J, were the ones that were expected to not have many differences because in all the mammals that they've already studied, it's been almost the same in all of them. So they didn't expect any difference or a large amount of difference between the inner neurons between the two. So this picture is showing the newborn giraffes uh, motor cortex. And this is where all these different neurons are that are over here. The pyramidal neurons, which is C and H, and as well as the dendrite segments, which is over here. And as you can tell, is very dark. And so these areas where you see the darkest uh, parts is where the dendrite activity was seen to be the most. Next is the one for the elephant, and in this, the inner neuron, which over here is G. So this is the inner neuron, and compared to the other inner neuron, they didn't expect much variety between the two. And the arrowheads that you see over here, those indicate the soma of the selected neurons that they used for this. And so all of this just represents the different types of neurons. So the flattened pyramidal neuron, F, which is over here, this you didn't see in the giraffe one, but however it's present in the elephant one. And also the inverted pyramidal neuron over here in E. Next, we have this colorful figure, and this is the neuron tracing in giraffes. Um, these blue ones over here show the pyramidal neurons, and this is the pillow surface, how much they stretch out or how long they were. And so all of these are the pyramidal neurons, A to H, which they were focused on. They also focused on I, the inner neuron, which is similar to the elephant ones, and J to N, these black ones over here, the extroverted neurons, as well as OQ, which are these purple ones, and those are the gigantopyramidal neurons. And so the arrowheads over here, those represent the axon from the neurons. So all these arrowheads that you see. sorry. So this is the same thing, but in the elephant instead. And in this, you see where it's showing you the flattened pyramidal neurons instead for A to C, which are these blue ones over here. Um, D to C is showing you, sorry, D to C, those are the causal neurons. And so in this, the elephant had the non-pyramidal multipolar neurons, which are the blue ones over here, which the giraffe didn't have. And this is just showing you all the different types of neurons that are on the pileal surface in in numbers. And again, the arrowheads represent the axons from the neuron. So from the study that was conducted and that they used the hypothesis to compare their results to, this is where they got this chart and were able to compare the newborn dendrites to the adult ones, and they were able to make a chart from this. So down here you see, sorry, you see the newborn pyramidal as well as the adult one, and you're able to see the difference between the inner neuron and the inner neuron and the inner neuron of the newborn and of the adult ones. So as you can see, all the structures of these are different from the newborn to the adult. And this is the neuron again that's expected to stay the same as stated in the hypothesis or have little variation. So results that were taken from this. So the results showed that the newborn elephant neurons were considerably larger in terms of all the dendrite and spine measurements as seen from the figures, and that the neurons in the newborn giraffes appeared more morphologically different compared to that of their adult counterparts. And the neurons uh, in the newborn elephant was considerably different from that observed in other mammals, which they've already seen in the adult elephant, and now they're seeing it again in the newborn elephant. So projections of these neurons that were observed in both these species show a larger variety in the elephant one. Like there were some neurons in the elephants that wasn't present in the giraffe one, like the flattened pyramidal neuron that wasn't seen in the giraffe one from the study, but it was seen in the elephant one. And the newborn projection neurons were smaller than the adult equivalents in both the species, which was expected to happen. And however, the inner neurons, which they didn't expect much change, these were about the same size as their adult counterparts. So, what's the hypothesis supported? This study did support the hypothesis that it came with, which are these three down here, and that were since they expected the dendrite system to be more extensive in the newborn elephants than the newborn giraffe, they were able to see that and prove that hypothesis right. And they also saw a more col- typical columnar exhibited in the giraffe neocortex than that of the elephant, as well as little variation in the neurons in both the newborn as well as the adult giraffe and elephants. And again, this is because the neurons that were already observed in the adults or in other mammals have been seen to be relatively uniform. So all parts of the hypothesis were supported from this experiment. So neurons allow the body to communicate, and studying the nervous system and the functions of it, as well as the different parts of it, advances our understanding of the basic biology of our body and the function of it. So knowing how things work can help shed light on what to do if they don't work, or they don't work how they're supposed to. This is in the body and in the body functions. And this can help researchers find ways to prevent problems that affect the body, nervous system, the brains, um, as well as different aspects of the brains, and how to better help fix that. Sorry. So a few things that can be taken away from this is that the animal with the most neurons is the African elephant, and they have about 257 billion neurons, and this is about 3 times that of the average human, because we have about 86 billion neurons. And so neurons are considerably large in terms of all dendritic and spine measurements. Um, in elephant neurons more than in giraffe ones, as well as the fact that dendrite remodeling occurs during developmental stages. So when the newborn is developing, they expect the dendrites to remodel themselves, but also in adult mammals as well. And this regulation can be can be regulated by that the neuronal activity that goes on in the brain. Thank you for watching my presentation.